Hi all, Group 3 will present on stronger gut microbiome modulatory effects by postbiotics than probiotics in a mouse colitis model. This is our outline, and now we'll start with the background. This paper involved the study of ulcerative colitis, which is a chronic inflammatory disorder of rectal mucosa, generally extending through part or whole of the colon. UC is commonly associated with epithelial barrier defects, dysregulated immune responses, and gut dysbiosis. Approximately 5 million people have been diagnosed with UC in 2023, and incident cases have been rising. Given its burden on the healthcare system, alternative disease management strategies have to be explored. UC is driven and maintained by a complex interplay of factors, and the gut microbiota is the main factor targeted in this paper. In the clinical setting, UC is commonly treated with oral corticosteroids and/or monoclonal antibody immunosuppressants. More recently, however, gut microbiota augmentation has become an emerging management strategy, with several studies involving the use of probiotics or postbiotics. Probiotics are defined as live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. Unfortunately, probiotics do pose safety concerns for certain groups of patients, as shown in the slide. In contrast, postbiotics is defined as a preparation of inanimate microorganisms and/or their components that confers a health benefit on the host. It has several advantages over probiotics. The authors recognize that there are existing studies on assessing the reduction in UC-associated symptoms. However, none feature an intra-study comparison between pro and postbiotics of the same strain. Furthermore, previous studies have shown that Bifidobacterium adolescentis as a probiotic is associated with a reduction in UC symptoms. However, B. adolescentis on UC as a postbiotic form remains unexplored, and importantly, the B8589 strain used in this paper has not been researched on.
Moving on to the study aims and design, there are two main aims of this study: to investigate and compare the effects of postbiotics and probiotics on disease condition, as well as their impacts on gut microbiome. Postbiotics and probiotics were derived from adolescentis B8589 strain. DSS model was used to simulate colitis. Some information on the DSS model is that DSS of 40 to 50 kDa in water stimulates colitis, most recapitulative of UC. The mice were subjected to the following conditions, with the first group acting as the negative control, second group acting as a positive control for UC conditions, and the last two as the treatment group for postbiotics and probiotics respectively. Next, the study used morphological and whole metagenome shotgun sequences as methods for data collection and analysis, details of which will be covered in the following sections. We now move on to the results and interpretation. From comparing the body weight change of different groups, treatment group did not show much difference from the positive control DSS colitis group. The team hence sought to use disease activity index, taking into consideration weight change, stool consistency, and fecal occult blood. There were some visible but non-significant differences between DSS colitis group and treatment groups. As colon distension is a hallmark of DSS colitis model, the team compared colon lengths of the groups. However, length of colon was not significantly different between treatment and DSS colitis group. Lastly, micrographs of the colon tissue sections and subsequent histological assessment revealed that postbiotics and probiotics allow recovery of the colon tissue to a state resembling the normal condition more than the DSS condition. This supports the stance that postbiotics and probiotics have positive impacts on improving UC disease condition. However, when comparing between postbiotics and probiotics groups, these four assessment tests revealed little to no differences between the two treatments on disease state.
Figure 2 looks at the microbial diversity of all four treatment groups. 2A and 2B uses the Shannon and Simpson index and 95% confidence level to analyze the alpha diversity. Across all four groups, there is no significant difference. The graphs are box and whiskers plot with a fatter width where there are more data points. So you can see that for the orange control group, there are two peaks with more data points at the second and first value of Shannon. After DSS intervention, the pink shape becomes very thin and variable. But despite this, postbiotic treatment can rescue the decrease in diversity after DSS induction. The blue shape starts to resemble the original control group. Moving on, 2C, E, and F explores the beta diversity using principal coordinate analysis. On the left, the graph compares DSS and control group to show that they are distinctly clustered. In the middle, postbiotic versus DSS graph shows a significant difference. The p-value for postbiotic versus DSS is less than 0.05. The value is 0.024. But this does not happen for the probiotic versus DSS graph on the right, where the p-value is 0.561.
Given postbiotics had a stronger capacity to regulate the gut microbiota diversity, the researchers now wanted to find out if postbiotics also have a stronger effect than probiotics in modulating the gut microbiota composition. Presented here are the core diagrams showing the composition of fecal microbiota at the phylum level on the left and the species level on the right across the 4 treatment groups. The upper half circle represents the 4 treatment groups, while the lower half circle represents the overall composition of fecal microbiota. In total, 8 phyla and 117 species were identified across the 4 treatment groups. While no significant difference was found at the phylum level between the DSS and postbiotic groups or between the DSS and the probiotic groups, significant difference was found at the species level. At the species level, both the postbiotic and the probiotic groups, in contrast to the DSS group, had similar effects in modulating species such as Escherichia coli. On top of the ones in common, the postbiotic group, in contrast to the DSS group, demonstrated additional ability to modulate more species such as Bacteroides intestinalis. Collectively, these results suggested that postbiotics had a stronger effect than probiotics on the gut microbiota composition in DSS-induced colitis mice.
The author revealed that both postbiotic and probiotic supplementation significantly impacted the gut microbiome functional potential. They used the human N2 pipeline to delve into microbial genes and functions, and the MetaSite database, which contained information about metabolic pathways and enzymes. They identified a total of 267 different metabolic pathways in the gut microbiome. Among these, an impressive 217 pathways were common across all groups. However, 15 unique pathways were specific to the DSS group, 5 to the probiotic group, and 2 to the postbiotic group. Their approach to validating these effects involved using the non-parametric Wilkinson rank sum test. The blue dot represents metabolic pathway that is significantly influenced, while the red dot indicates non-significant differences. This led them to identify 40 differential metabolic pathways when comparing the DSS and control groups. By comparing the DSS and probiotic groups, they observed 61 differential metabolic pathways. When comparing the DSS and postbiotic groups, they uncovered a staggering 144 differential metabolic pathways. Therefore, postbiotic could modulate the functional potential of the gut microbiome to a greater extent than probiotic. So here's a quick summary.
A correlational analysis was also performed to compare any intergroup changes between the postbiotic and probiotic fecal microbiota and their associated metabolic pathways. The results of the analysis are displayed in the heat maps below. It was discovered that postbiotics and probiotics mediated divergent changes in the intergroup associations between fecal microbiota and metabolic pathways encoded by them. In the postbiotic group, most of the 20 metabolic pathways were upregulated by the 2 bacteria highlighted. Additionally, there was also significant downregulation of the metabolic pathways. In comparison, the probiotic group generally displayed fewer and weaker correlations between the fecal microbiota and the encoded metabolic pathways. This suggests postbiotics had a greater regulatory effect on the metabolic pathways than probiotics. Interestingly, of the top 20 bacterial species identified in each group, 16 were in common. 7 of these showed incongruent correlations with the top 20 metabolic pathways, suggesting the postbiotics and probiotics may confer opposing regulatory effects on fecal microbiota. Some of the limitations of the study are as follows. Firstly, the mechanisms behind the metabolic pathways of the postbiotic and probiotic strains are unclear. Secondly, although mice showed a deviation from a diseased phenotype, this doesn't imply a healthier state had been attained. And finally, a mouse model was used to infer the effects of postbiotics rather than an organism more closely related to humans. Future work for this study could involve clarification of metabolic pathways that contribute to ulcerative colitis pathogenesis. Moreover, having additional experimental groups could better elucidate the postbiotic effects. And lastly, postbiotics and probiotics' ability to prevent ulcerative colitis should be investigated. Thank you for listening to our presentation.